Then I'm happy to introduce our next speaker, Dr. Max Crüsemann. Max did his PhD with Björn here in Bonn. He worked on biosynthesis, and you already mentioned the homomycin, for example. He then joined us in San Diego for postdoc with Brett Moore. But he also worked a lot on MS networking and the structure elucidation of nature products and therefores. Now he's back in Bonn and is there a group leader working with biosynthesis of nature products, and I think we'll talk about his work right now.
Exactly. Thank you, Nadine, for the nice introduction, and thank you, Martin and Megan, for the invitation and for the great meeting. It's really an honor to speak here about my favorite natural product, actually, which is a G protein inhibitor. You can see the structure here. And more specifically, I would like to talk about thioesterase domain that does a really important modification and unusual modification in the biosynthesis of this compound. So, it's a G protein inhibitor, and so I would like to talk a little bit about this quite unusual mechanism of action here. So G proteins are coupled, as the name says, to G protein coupled receptors, which are much better known actually, and these sit in the membranes, are really ubiquitous, I would say, and basically shuttle signals from outside the cell into the cell. Then the G protein sits at the inner membrane of the cell and does signal transduction and basically then initiates the signaling cascades in the cells. And there are various reactions. So this is a really important function. We have these three subunits here. As I said, GPCRs are really crucial for all of us, for really for important functions. And thus it's clear that many drugs also target these GPCRs. And so our natural product here, FR 90039, which I will from now on just call FR, really does a very good job in inhibiting these G proteins, specifically GQ proteins, which is an important family of G proteins. So it really blocks the G protein and does it in a very selective and potent manner. So nanomolar potency. And so 10 years ago, the crystal structure of an analog of FR, another cryptic name, YM025490, just short YM, was elucidated. And there you can see that that the natural product really goes into the C-inch region here, this linker region, where the nucleotide exchange is conducted and really blocks this protein in a very very smart way. And so you can see here that FR and YM are very closely related, so they just differ in a few method groups. And so this is a really nice mode of action here. And it's important to say concerning the mode of action that these G proteins, they are not much less G proteins than GPCRs. So there are like 800 GPCRs now, a really big variety, but just 4 families with a few subfamilies of G proteins are known. So in the evolution, there was a really expansion of GPCRs. You can see it here in this picture. And the message is that these G proteins did not diverge so much. So that means that they have like a barcode, which is very rigid. And so if you inhibit these G proteins, you can block many receptors at one time. And so this is a very effective strategy from nature for a toxin. And so we in Bonn, we study these compounds for quite some time now, for a few years now. And due to their unique mode of action, they can really applied as pharmacological tools. So pharmacologists really love these compounds because they can selectively switch off pathways and study the signal transduction. And you can see here on the slide that in the last years there has been kind of explosion in the use of these compounds, which was reflected in the citations since 2015, when you can see that this was called the resurrection of FR and basically the characterization as a pharmacological tool. Because the other compound YM was commercialized by a Japanese company and was not available to the public anymore. And so, yeah, these compounds are really, really nice tools for the pharmacologist. And in addition, they may have some medicinal relevance as well, which we are studying in Bonn as well, because there are, of course, many diseases are G protein related. For example, this asthma, COPD, this lung diseases. Here we, if you apply FR as an aerosol, inhale it, it has pretty good effects, even better than the drugs that are on the market. And then there's another recent example. There's the uveal melanoma. It's a progressive eye cancer, and we could show, or our colleagues could show, that FR actually reduces the tumor size. So this is a pretty, pretty nice effect in this special kind of cancer. So this is a, as you can see, it's a really interesting and promising compound from a pharmacological point of view, medicine, but also for natural products researcher like me, because this compound is quite rare. It has been, this family has been isolated a few times, just a few times, but in many very different ecosystems. So you can see here that it has been isolated from soil, from plant leaves, which I'm going to talk about a little more in a second, and also from the marine environment, from a tunicate a few years ago. So it must have, or we assume that it has a quite, it has a relevance ecologically. And so first it was isolated from an Asian plant, Alesia crenata, which you can also buy in garden centers, quite nice, beautiful plant, and specifically it was isolated from the leaves of this plant back in 1988. And then when we started working on the project, we looked at the distribution of this compound in the leaves, and could see that it was mainly concentrated in the leaf nodules, the outer part of the leaf. This was done in Peel's collaboration with Jan Peel. And so we assumed, okay, this there must be the producer, because we assumed the bacterial origin for this compound. We could see that FR is really toxic to many organisms there. So when you take it up orally, you can see a really significant reduction of blood pressure, and also insect G proteins are, I would say, equally inhibited by this compound, very, very potent. Since these G proteins are, G-cupins are conserved in many, many organisms. And also insects, if you feed this compound to insects, they will die after a few days, concentration dependent. So, yeah, we thought, or we hypothesized that FR protects the plant from these predators. And if you now look closer at these leaf nodules, actually a few years in Ebert's lab and Clavier, they discovered that there actually are bacteria in these leaf nodules that are endosymbionts. And these endosymbionts are Bocardieria bacteria, and they were called Bocardieria crenata, and then afterwards the genome sequence. And actually, it's one of the smallest genomes that were known at the times, or at least of this genus. So very, very small, very eroded DNA. Many pathways were just destroyed, but two secondary metabolite pathways are present on the plasmids, and one of them was NRPS, which we assumed that this was responsible for the production of FR. So now let's have a closer look at the structure of FR. It's a cyclic dexipeptide. It's quite complex. Now with 8 residues, none of them are, one of them is proteinogenic, the alanine, and many of the others are highly modified. So we have 3 times this hydroxyleucine here, which is in black, and 2 times of them acylated. We have we have dehydroalanines, which we've also seen in the previous talk by Elke, but also quite rare. We have this dimethylated threonine and a few others. And also conformation, we have 2 cis peptide bonds, so it's a kind of a special conformation here. So then we want to look for analogs in the plant leaves. And so we did some molecular networking study and look for analogs here by GNPS. And okay, with FR is the main compound, but you can see the red compounds here. We have found, and Rafael has isolated some analogs that mostly vary in the side chains here, the acyl chains, I would have to say. So there's variation in the acyl chains and not much else. And then there are some other compounds seen in the network, which we couldn't isolate, but could analyze them by mass spec. And these are much smaller than the FR. And from MS analysis, we deduced that this could be the FR core, basically just this macrocycle here. So I forgot to mention that there's a side chain, and we were assumed that this has been missing in these compounds, but yeah, we couldn't do further work with them. And also here we found many variations, so the same variations basically in these smaller metabolites. And when we did bioactivity tests on these compounds, we saw that the analogs are have almost the same activity as FR. But then some other studies were done which synthesized analogs synthetically, and many of these analogs, most of them, really reduced the activity of FR. So basically, we could deduce from these studies, also from some modeling, that the biggest part of the natural product is the pharmacophore, and you can only change a few positions here to not lose activity. So it's basically the natural product is optimal for binding GPU. And so now, of course, we analyzed the biosynthetic gene cluster. We were interested in it, and we have here, interestingly, 2 NRPS systems. So we have a heptamodular NRPS, 7 modules that we suggested to produce this FR core, which we have just seen in traces, and then we have a monomodular NRPS, FRSA, which we thought could synthesize the side chain. And then maybe through this transesterification, attach it to the FR core to make the product FR. It's a quite compact gene cluster. We have just 8 genes, 5 NRPS genes, and just 3 other genes. Yeah, and so we have an MBTH-like protein, and then there are just 2 other modifying enzymes, hydroxylates, FRSH, which installs hydroxy groups, as we thought, and FRSC, which reduces pyruvate phenolactate. And all the other modifications are encoded in the NRPS system. So we have 4 methylation domains, we have C-starter domains that perform the acylation. We also think that the dehydration is encoded in the NRPS system. So that's to do biosynthesis. And then I actually was able to heterologically express the gene cluster in E.coli, but only after lots of work and modifications with chaperones, and then produced CFR but at very, very low yields. And I tried a lot of things to improve it, but it was not really working. So with this system, we couldn't really do in vivo studies at that time. And so that's basically where the story I'm going to tell you now starts. Because we started looking out for cultivable producers that might be easier to study. So this is the Procalarian gene cluster. And then, yeah, I started looking initially for other sequences that might code FR biosynthesis. And then René Richards came to the lab also, and We saw just some fragments, and he was able to assemble these fragments. It was a chromobacterium, and then we was able to assemble the gene cluster, and basically it looks exactly the same as in the endosymbiont, just a little bit higher GC, but the rest is, I would say, more or less identical from the architectural point of view. And this bacterium, this chromobacterium, this is really nice because it also makes FR very rapidly and also in good yields, and we can cultivate in the lab just very easily. So this was great because now we could start studying the biosynthesis in more detail. And now let's go back to the biosynthesis. Now we wanted to study especially the side chain biosynthesis and this attachment of the side chain to the metabolite. And we thought, okay, probably it's the TE domain that could perform this attachment, this intermolecular transesterification. Maybe one little note, when we look at the sequences of FRSA, it was very, very similar to the FRSD, which is the first module of the big NLPS. We'll come to that later at the end of my talk. And so then Cornelia Hermes, a grad student in our lab, started investigating this side chain biosynthesis in vitro, and she could clone the whole module, also some of the domains that Daniel cloned the hydroxylase, FRSH, which was also needed to produce this propyl hydroxyleucine. And so our theory was that first leucine is activated onto a PCP domain, and then hydroxylation takes place. This is quite a not very well investigated monooxygenase. There's just one member from chloramphenicol biosynthesis, and also some glycopeptides, homologous proteins like it. And then finally, the C domain was predicted to attach the propyl group. And so she developed an in vitro assay in the reaction tube, where basically all the precursors and cofactors were added. And after some time in hydrolysis, she thought she could detect propyl hydroxyleucine. And indeed, we were lucky or happy to see that indeed the side chain is produced in vitro in comparison to the standard that we also synthesized. So this was great. But now to study the thioesterase domain, we of course needed a substrate for this domain. And if this model is really right, then if you would then knock out this FRSH, maybe we could see accumulation of this intermediate FR4, we were hoping. And so we did this, René Richards did this, and indeed, when you knock out FRSA, you can see that much more FR4, which was before produced in very small amounts, was seen. So this gave us a chance to isolate the metabolite. And then we saw, you might have noticed that the chromobacterium is purple. Yeah, this is due to a pigment biolacine, which is produced in many of the chromobacteria. And when he knocked out the biolacine, the yields of both FR and just the first mutant, and in the double mutant, the yields of FR core, you can see in the lowest trace, really rose up. So this is a really nice system to even better study biosynthesis. And so now we were able to isolate FR core and confirm its structure by NMR. Now we had a substrate, everything in hand to study the thioesterase reaction. And now we can take a little bit closer look at this intermolecular transesterification. This is quite rare, unusual reaction in nature. There have just been a few other cases where this has been studied. In the case of salinamide and Brad-Moorstead, where I was also working at the time, this was the first instance. And then necroxine, PFAS and its hybrid that was isolated this year in St. Patrick's, that there are also such a transesterification, and basically the extension of the molecule suggested. So basically you can, the goal or the mechanism is that this another moiety is attached to an already existing compound. And so now we want to study it for our FR. And for MegaSyn have 3 different assays with all the substrates. So just with a TE, with a SN substrate that was synthesized in Gütschlow's lab and our colleagues, and of course FR core as substrate. The second assay consisted of the full module, the SN substrate and FR core, and in the third setup, we did the whole in vitro biosynthesis. That means with the module and with just the precursors. And we were really happy to see that in all 3 cases we could produce FR in an epi, in a reaction tube. Actually, just the TE and the SN yields were the highest, so this gives us also some maybe opportunities to do some engineering or some enzymatic synthesis of analogs. And then we also added some variations to the assay. So we added shorter acyl chains, acetyl-CoA. You were able to see the formation of the FR core, which is a derivative, and even the longer side chain was produced. The butyryl side chain was also well tolerated in vitro and also in vivo, and we could elucidate this novel metabolite, butyryl FA. It's a novel FR derivative. So that was really nice. And then she went a step further. I told you that FRSA and FRSD, the 2 modules, are very highly similar. And so what happens if we exchange FRSA, CATs, and FRSD and try to perform the TE assay with the FRSD module? And indeed, we can also make FR with this setup. And interestingly, now the ratio of the products changes now. The acetyl derivative FR2 is produced in higher amounts than the FR, which implies that the C domain, the sub-C domain, has changed its substrate specificity for shorter acid chains, which is also reflected in the natural analogs. So this position you usually find acetyl groups. Then we were interested in asking, okay, what is actually the benefit of this modification? So, what is the effect of the side chain? And we now that we had the FR core, the derivative without side chain in hand, we could perform comparative bioactivity tests of FR and FR core. And so here we did it on the human GQ protein, and we could see that FR is 14 times more active than FR core, which is the really big difference. And the affinity of the compound to the G protein is even 200 times higher. So the side chain has a really big impact for the bioactivity of the molecule. Yeah, almost crucial. And then we also fed FR and FR core to insects again, to these stink bugs here. And also here we could see a really big difference. So FR is, as we see in the beginning, is really toxic. After a few days, our animals die. But FR core, in the highest dose, okay, the animals die as well. But in the medium dose, the animals do not die. So that means that FR, the phylogenetic is much more effective with the side chain. We also modeled FR and FR core to the target, to the GQ protein. Here, the side chain is here in green. And if you compare these models, they sit at the same position almost, but the side chain has really many additional interactions with the binding pocket. And also, it has some intramolecular effects, so it stabilizes the conformation. So here we could really see that the interaction with the target is improved. All right, so finally, we thought, okay, FR side chain makes really a big difference, but what is maybe the evolution, or could we get some insights into the evolution of this system, of this modification? So now we have the problem that we just had 2 FR VGCs, and they're also identical. So we learned that we cannot perform this really evolutionary studies that Najim, for example, with the glycopeptides has presented, where you could really compare the classes because they were identical. We just said 2. But also, we did not see really big similarities to other VGCs, but maybe we had missed something, so maybe we wanted to look more into the relations. And maybe, at least from some bioinformatic analysis, we could get some insights into the evolution of such. And so we performed, or we used the great tools Manix has presented yesterday, and we did try to do global NLPS analysis with as many NLPS systems as possible to see the relations of FR to other NLPS. But we very quickly realized that this is very time consuming, as Manix pointed out, and it just did not stop processing this analysis. And so then we figured that this novel novel invention, big slice, is much faster, and you can do even more global analysis with this program. And we figured also out that this big time database, this is 1.2 million VGCs that Manix had presented yesterday. And when we looked for our FR gene cluster in this database, there was already one deposited in the myelin, we could see that our gene cluster was indeed in one of these families with 220 VGCs. But you also pointed out that at this stage the relations are not that clear. So we took these 222 VGCs and performed then a big scale analysis again with these gene clusters. And there we can see that if you use a very, very high cutoff, or really high cutoff at 0.6, which I believe is really high, and most of these 200 gene clusters cluster together, but the FR are still separate. So that means I would say that indeed the FR gene clusters are pretty distinct from other NLPS VGCs, and they are not really closely related NLPS in the database at the moment. Now let's go back to the bioinformatic analysis of FRSA. I told you that big parts of the sequence are identical, especially this sequence stretch here. It's like 2,600 nucleotides, including the whole A domain, biggest part of the starter C domain, and also some parts of the TE domain. We also see that the other A domain for the third leucine in the molecule is also identical. So here, this stretch, we see really the same sequence 3 times in the gene cluster. So this points, I would say, to a duplication event during evolution and to an intra-cluster evolution, since we also do not see similar sequences in the database. We then performed the phylogenetic tree of the starter C domains, since they were a little bit different, at least a little bit. And we took a lot of sequences here from unrelated starter C domains. When you look at this tree, you can see that this FR, FRSA, FRSD C domains, of course, very similar to each other, but then move quite deeply in the tree, so there are not other very similar C starter domains. So, of course, they are related to each other. And lastly, we were investigating the thioesterase domain, the evolution of this transesterifying FRS-ATE, and luckily, 2 years ago, this TE domain tree was published. Also, it was here on the poster at this conference by Untitled Chapter. And basically used this tree as a basis and just added our TE domains and some others that also might perform this transesterification to this tree, and we can see that our TE domains cluster very closely with the FRSG TE domains, which are at the end of our cluster and make this macrocycle. And also then the other transesterification also clusters quite closely. So there might be an evolutionary relationship between FRSA and FRSG domains. But I think at the moment we cannot say much more. So now I would like to come to the conclusions of my talk. So FR is a really strong and selective GQ inhibitor with some relevance. We could find it a cultivated FR producer. We could see that two NRPS systems cooperate during FR biosynthesis. We have this unusual TE domain which performs this transesterification of the side chain. The side chain is really important for GQ inhibition. And in the end, we can probably conclude that FRs so far really rare and uniform gene cluster family that shows elements of duplication, so maybe like an intra-cluster evolution. And one could speculate that FR4, the lesser active metabolite, was maybe there before as an ancestor, and maybe due to duplications and evolutions, the side chain has been generated. But yeah, so far it's just speculation, and we don't have any evidence for it. So now I'm at the end of my talk. I have to thank many people, especially first the people who did the work. Cornelia Hermes, grad student, did very nice work on in vitro. René Richards was in vivo work, postdoc, and of course other people in the lab in our group, collaborators at the Bonn. I especially have to thank Professor Gabi König for all the support and mentoring and the great teamwork, and of course all other collaborators. Yeah, thank you for your attention and happy to take questions.
Thank you, Max. Nice comprehensive story. Questions for Max? Maybe I start. So you showed that there's this subcluster that makes a side chain and the other cluster that's FR4. So did you do some kind of bioinformatic analysis, sequence analysis to look specifically for only parts of these, like either the first or the second part? And then you showed the big scape analysis, where in big fem you have all these 200 clusters together. Did you look at these clusters and why they would network together with the FR cluster?
So maybe the second question first. These are very diverse. So we saw that there were also some known clusters. I mean, they were all NRPS, but you could not see any obvious relationship. And so basically, we looked at different cutoffs, and the picture showed you that at some point they cluster together. Of course, I mean the higher you make the cutoff, then of course there are some relationships because they're NRPS, but still, I think at the usual cutoff points they are really more separated, and FR also at this high cutoff shows not really similarities to that. Maybe to the first question, I mean, we compare the sequences in the cluster, and we see these identical sequences, and yeah, we also see it at some other points. So, but nothing really similar outside the cluster. So this points to maybe that there will be some duplication events in the gene cluster.
Have you also tested the vitro side chain analog provided?
Yeah, so the question was about the vitro. Also the slide for that. Interestingly, the vitro analog was also lesser active than FR, 7 times less active, which is a quite big difference, just concerning we have only one methyl group. So it seems that also the side chain, you cannot do many variations. So since the activity is so high, it seems to be pretty optimal, and yeah, only this longer chain seems to have a negative impact on the bioactivity.
I have another question because I think it's really fascinating because there's a huge amount of data and really you showed that there's no big similarity to other gene clusters. It seems like, did you check metagenomes? Because I think if these are really these molecules have this important role on the leaf to really be an insect deterrent that you suggested, right? Then you would think you would find it in more bacteria.
Yeah, you're right. I mean, we would like to do some maybe metagenome mining. That would be great. Also thought about that. Maybe since we also see the different contexts, not only in leaves but also in soil. We haven't done that. We would like to do it. I mean, so far we just looked at all the sequences that are in the database, and there we didn't see anything. So, but of course there are many libraries that could contain have more clusters, and I'm sure there are somewhere. We just have to find them.
Yeah,
I have maybe a similar question. So the binding site of FR is quite unique, right? So have you, or has anyone tried to look into this binding site, whether it fits in silico additional natural products?
I think this has been done. I mean, there have been screens with a lot of compounds, synthetic and natural compounds, and nothing really came close. I'm not aware of a specific in silico screen. Basically, we didn't do that because our compound was already working pretty well, so I think so far there was no need to look for better ones. But this would be maybe a good way to find related compounds, at least for mode of action.
I'm just surprised to see that in such an important target, there's just one natural product. It's hard to believe.
Yeah, that's true. I mean, this is really, I think it's really special compound so far, but you're right. I mean, this is, of course, there's opportunity, or there could be something that also binds, and maybe not that specific. Maybe that's if it's more simpler compound. You could you also bind to other G proteins. In this case, we only bind to GQ, which is quite special, I would say.
If there's no more further questions, let's thank our speakers again.